Hello everyone, let's get started. Today we're going to be talking about membranes. We're going to start off talking about membrane structure and function, and then we'll move into the mechanisms of membrane transport. Now, a lot of what we'll talk about today is review from BIO312 Molecules and Cells. We'll go through that material to refresh it in your memory, and then we'll add some more detail to what you already know as we learn more and more about this critically important structure, not only around every cell, but around every organelle as well. Let's first take a minute and review all of the functions that cell membranes have. Remember, first and foremost, what a membrane is doing is creating a compartment. The membrane that wraps around every cell is defining the space that is that cell. It's separating what's inside the cell from what's outside in the environment beyond it. The same thing's happening around organelles. Inside of every eukaryotic cell, there are several different types of organelles, as we've discussed, and there is membrane that surrounds those organelles. That membrane is separating out what's happening inside the organelle from what else is going on in the cytoplasm of the cell. So first and foremost, membranes define boundaries. Membranes are also playing a critical role in determining what can come in and what can go out of a cell or an organelle. And again, that's really important because that allows the internal environment to be different from what's outside of it. The concentration of certain molecules can be very different inside a cell compared to the external environment, or inside an organelle compared to the rest of a cell. Membranes are also loaded with protein, and the protein plays a critical role again in helping things get across that membrane or in blocking them from getting across. There are protein channels, protein carriers, and protein pumps that are doing this important work. Membranes contain other proteins that function as receptors. And remember, what a receptor is doing is receiving information from outside of the cell. Receptors are going to interact with other molecules, and that interaction will then set into motion some kind of an effect in the cell or in the organelle. Membranes contain enzymes as well, embedded directly into the bilayer. Enzymes that, again, when they interact with other molecules, are going to set into motion the kinds of chemical reactions that are critical for the functioning of the cell. And finally, there are proteins embedded in membrane that play a role in helping cells adhere to each other. These are the different kinds of cellular junctions that pull cells together and hold them together in the creation of tissue in an organism. So, yes, cells are surrounded with a membrane. That's what helps define the boundary of a cell. But cells are also full of membrane internally. Remember, even though we sometimes draw cells as having mostly just a lot of empty space inside of them, in reality, when we start to examine cells underneath an electron microscope, we see that with all of the organelles that are inside of a living cell, there really is very little empty space. In fact, most of the space inside a cell is full of membrane that surrounds all of the different types of organelles in that cell. each of the membranes that surround each different type of organelle, remember, are going to be different from each other because each organelle has a different function. So the permeability of the membranes that surround different organelles is going to be different one to the next. The amount of protein, the type of protein, it's going to vary from membrane type to membrane type to membrane type. But the important thing to remember is that when you look inside of a living eukaryotic cell, most of what you're going to see is membrane. And you can see that depicted in the image on this slide. We're looking at, again, an electron microscope image of a eukaryotic cell, of a portion of a eukaryotic cell. And you can really see just the vast amount of membrane, of surface area of membrane inside the cell. In the lower right-hand corner, right here, you're looking at the nucleus of the cell. So again, we can't see the entire nucleus here, but we can see the bulk of it. The nuclear envelope would be located right here. You can see varying shades of gray all the way to black inside of this nucleus. You can see this large dark area here. This would be a nucleolus. And all of the chromatin material is packed into this nucleus inside the center of the cell. Then if you go outside of the nucleus and you look carefully, you can see all of these lines. And all of these lines are sort of twisting and turning and just filling up most of the space inside this cell. This turns out to be endoplasmic reticulum. Most of it is rough ER, although that's hard to see at this magnification. There are some circular, vesicular organelles visible as well. Remember, some of those might be lysosomes, they could be peroxisomes, but the bulk of the membrane that you're looking at is endoplasmic reticulum. That tells you something about this cell. If the cell membrane is located here, and again, we're only looking at a portion of this cell in this image, the vast majority of the space in that cell is full of membrane, and it happens to be endoplasmic reticulum membrane, which tells you something. It tells you that the production of protein in this cell is tremendously high. There's lots and lots and lots of protein synthesis going on in this cell. So let's take a step back now and review the general structure of the bilayer that is a cell membrane. Remember, when you look at any membrane, whether it's a plasma membrane around the boundary of a cell or the membrane that is wrapped around different organelles, it's primarily composed of two types of macromolecule, lipid and protein. In fact, if you were to separate out the different types of molecules in a membrane, the amount of lipid, specifically phospholipid and sterol, would be about the same as the amount of protein. So those are the two types of macromolecules that make up the bulk of a membrane. And of course, the membrane is made of a bilayer of phospholipid. Two layers of phospholipid molecules that associate with each other because of their chemistry. Phospholipids, one of the three kinds of lipid, phospholipids are amphipathic. They have both hydrophilic and hydrophobic regions within one molecule. So they have areas that are polar and areas that are non-polar. Remember, the proteins that are embedded in membranes are also amphipathic. They have regions that are polar, regions that are non-polar. Phospholipids will assemble into this membrane bilayer spontaneously in the watery environment that is the inside of a cell. Individual phospholipids are not covalently bound to each other in a membrane. They are held together by weak attractions, by hydrogen bonding between the polar regions and van der Waals interactions in the non-polar regions. And remember, in a phospholipid, the polar regions are the heads and the non-polar regions are the tails. And that's what we're looking at down in this image at the bottom of the slide. You can see a bunch of individual phospholipids that have come together and spontaneously assembled into a bilayer. We're inside a cell, we're in a watery environment here. So perhaps the outside of the cell is over here, the extracellular space, and the inside of the cell is over here, the cytoplasm. And here are the phospholipids that make up this little piece, this little sliver of the membrane around this cell. The circular areas represent the heads of the phospholipids, and those are polar. Those are hydrophilic. The squiggly lines underneath each one of the heads, these represent the tails. These are hydrophobic or non-polar. So all of the tails are staying together. They're drawn to each other and they are held together with weak interactions called van der Waals interactions. In comparison, the heads are all oriented towards the water because the heads are polar or hydrophilic, water-loving. So the heads are oriented towards the water that is outside the cell, and the heads are oriented towards the water inside the cell, in the cytoplasm. This is a spontaneously forming structure in a cell. Phospholipids, again, are one of the three types of lipid that we find in living cells. Remember, there are phospholipids, there are fats or triglycerides, and there are sterols. The phospholipids are the most abundant type of membrane lipid. The head of the phospholipid has two components in it. It has a glycerol molecule and a phosphate group. Sometimes the heads of phospholipids have additional, what we call small molecules attached to them. We're going to talk a little bit more about those extra groups in a minute. But for now, just know that every phospholipid has a glycerol and a phosphate in its head region, and some of them have additional molecules attached. Now the tails, there's actually a pair of them. The tails are made out of fatty acids. And fatty acids also have two important components to them. They have a hydrocarbon, a chain of carbon and hydrogen atoms bound together, and at one end, a carboxyl group. It's important to remember that a fatty acid and a hydrocarbon are not the same thing. Fatty acids contain hydrocarbons, but they also contain these important carboxyl groups. In fact, it's the carboxyl group that attaches the tails of a phospholipid to the glycerol molecule. And we'll take a look at this in just a minute. Now, when we look at the phospholipids across different types of organisms in the three domains, we can see some variation in this structure. So there is an exception to the anatomy of a phospholipid, and that exception is a molecule called sphingomyelin. In sphingomyelin, the phospholipid contains something called sphingosine in place of glycerol. And again, we'll take a look at this in just a minute. But understand that, well, the vast majority of phospholipids contain a glycerol and a phosphate in the head region, and two fatty acids as tails. There is an exception to that anatomy in this molecule called sphingomyelin. Sphingomyelin doesn't have glycerol in its head. It has a molecule called sphingosine instead. Now, the other membrane lipids that we find include sterols, and that includes molecules like cholesterol, and also a molecule called a glycolipid. As we'll see in just a minute, there are a good number of phospholipids that are modified to contain a short chain of carbohydrate. Remember, when you look at a term and it starts with glyco, that glyco should remind you of carbohydrate. So a glycolipid is a lipid that's been modified to contain some carbohydrate. Some membrane phospholipids have carbohydrate chains attached to the head region, and so they are now converted into what we would call a glycolipid. Let's take a look at some of this structure now. On this slide, you're looking at three different representations of a phospholipid. Over on the right-hand side, this is the kind of depiction that shows the individual atoms of the phospholipid as different colored circles. So as we've seen in other lectures, the red circle represents oxygen, the black circle represents carbon, the white circle represents hydrogen, and so on. This area up here represents the head of the phospholipid, and down here you can see two fatty acids, or the tails. Now, if you look at the center image, now we're looking at a more traditional depiction of the structure of the phospholipid, because you can see all of the individual carbons and hydrogens and oxygens and phosphorus and so on throughout the molecule. So in this image here, that orange type color structure in the center here, this is glycerol. The green color represents the phosphate group, and the blue color represents one of those extra small molecules I mentioned in the last slide. This is a molecule that happens to be called choline. And you can see again, it's attached to the head region of the phospholipid right at the phosphate group. We'll talk in just a minute about the importance of these extra small molecules that sometimes get attached to phospholipids. So the glycerol and the phosphate group, and also this extra choline molecule, these make up the head region of the phospholipid. This is the polar region. This is the hydrophilic region of the molecule. Now, these are the tails. These are the two fatty acids that represent the tails of the phospholipid. If you look up near the glycerol, right here and right here, these are those all-important carboxyl groups. All right, there's a carboxyl group on each tail, right at the point where the tail connects to the glycerol molecule. The carboxyl group is there to give reactivity to that fatty acid, to allow it to interact with and react with the glycerol and bind covalently to the glycerol. The rest of the tail has a very simple structure. It's all carbon and hydrogen. This is what we mean by a hydrocarbon. So this is a hydrocarbon and this is a hydrocarbon. Here's the carboxyl group. So together, the hydrocarbon and the carboxyl group make up a fatty acid. So this area of a phospholipid, because it is loaded with carbon and hydrogen, it is non-polar. Remember, carbon and hydrogen have the lowest electronegativity of the four essential elements that we've discussed. Carbon and hydrogen, for all intents and purposes, have the same low electronegativity. So they are going to share electron pairs equally in the bonds that connect the carbon and the hydrogen atoms together. So all of this equal symmetrical sharing of electrons creates a non-polar chemistry, a hydrophobic chemistry, the tail region. Now, the other thing that's very evident in this drawing is that this tail is pin straight and this tail is crooked. This tail has a kink in it. And they're showing us that there's a carbon right here, covalently bound with a carbon here, not with a single covalent bond, but with a double covalent bond. And if you recall, when you see double bonds in the fatty acid tails of a phospholipid, those double bonds create these kinks. And these kinks are quite stable. They form where the double bond is, and structurally they stay very still. So this space that gets opened up changes the permeability of the membrane in this area. It opens up some space in the tail region of the membrane and makes it easier for things to get across the membrane. We'll talk more about that in a few minutes. The last drawing over on the left-hand side of this slide is just a very simple depiction of the individual molecules that come together to create the phospholipid. They're showing up at the top the three structures that make up the head, which is glycerol, phosphate, and in this instance, an extra molecule called choline. Down at the bottom, they're showing us the non-polar or hydrophobic tails that are made primarily of hydrocarbons, but have a carboxyl group up at the top. Together, this makes these gray colored structures fatty acids. So hopefully that was all review for you, and having talked through it, we've brushed some of the cobwebs away from your memory of what phospholipid structure looks like. I said at the beginning of the lecture that one of the things we're going to do today is learn some new detail on top of what you already know about membranes, and that's what this slide is about. It turns out that while the phospholipid molecule is primarily the same throughout the membrane, there are some structural differences between phospholipids, and it's all dependent upon that small extra molecule that gets attached to the head, like the choline we just looked at. So on this slide, you're looking at 1, 2, 3, 4, 5 different phospholipids. You're looking at the two fatty acids depicted in gray. Here's that carboxyl group at the top of the fatty acid. Here's the glycerol. Here's the phosphate group. And then in pink or magenta up at the top, these are those extra small molecules that are sometimes added to phospholipids to slightly change their chemistry. In this instance right here on the left-hand side, we're looking at a molecule called ethanolamine. This molecule right here is called serine. This one here is called choline. And this one over here we'll save for just a minute. So these three, 1, 2, 3, these are 3 phospholipids. They have the general structure that all phospholipids have: glycerol, phosphate, and 2 fatty acids. but they've been modified with these extra groups. So that now we can call this molecule phosphatidyl ethanolamine. And we can call this molecule phosphatidyl serine. This one in the center is phosphatidyl choline. All three of them are phospholipids, but they all have slightly different chemistry, and that's because of these additional groups that have been added. Now, I mentioned this molecule called sphingomyelin a minute ago. I told you that sometimes the anatomy of a phospholipid is a little bit different than we normally expect to see, and the example is this one called sphingomyelin. If you just briefly look at this molecule, it looks a lot like, in fact, this one, doesn't it? It has that same choline molecule up at the top. It looks like it's got a glycerol and a phosphate and two fatty acids. But if you look more closely, you realize that this central area right here is different from this area right here. This does not have a glycerol. This molecule right here is not glycerol. Instead, it's a molecule called sphingosine, and you can see sphingosine all by itself right here. So we don't have the classic structure of a phospholipid here. We don't have glycerol, phosphate group, two fatty acids. Instead, we have a sphingosine, a phosphate group, and one fatty acid. Because you see, sphingosine already has a structure within it that looks like a fatty acid. So when we bring sphingosine in to replace the glycerol, we're also replacing one of the fatty acids. So sphingomyelin is a very unique molecule in that it looks almost exactly like a phospholipid, but it's not. It's a variation on a phospholipid that functions in a very similar way. Now, why do we care about this? Why do we care about the fact that some phospholipids have these little extra groups attached to them? Why do we care that there's a molecule called sphingomyelin that, for all intents and purposes, looks like a phospholipid, but is structurally different from a traditional phospholipid? Well, the reason we care is because these variations, these structural variations, allow for the composition of lipids inside of membranes to vary quite a bit from species to species and from domain to domain. If you look at this slide down at the bottom, you can see a combination of graphs. And what this image is showing us is some of the differences in the membranes among different organisms. What's different from organism to organism to organism is the percentage composition of the different varieties of phospholipid. For example, anywhere you see a red bar, you're looking at that variety of phospholipid called phosphatidyl ethanolamine. Everywhere you see an orange bar, you're looking at phosphatidyl choline. Everywhere you see this sort of maroon colored brownish bar, that's phosphatidyl serine. The teal colored bar is our friend sphingomyelin. And there's even a couple of other types of phospholipid we haven't mentioned yet. Phosphatidyl inositol, phosphatidyl glycerol, and another one called diphosphatidylglycerol. glycerol. All of these names are not super important for us to know. What's important for us to know is this. Look at the difference in the bars as we travel across from organism to organism to organism. This first group of bars represents the composition of the different types of phospholipid in the membrane of the cells inside a rat's liver. So we're looking at individual hepatocytes or liver cells that come from a rat. Rats are, of course, multicellular eukaryotic organisms, and their membrane, at least around their hepatocytes, contains a wide range of different varieties of phospholipid. It even contains that strange variation called sphingomyelin. There's a variety of different phospholipids surrounding this cell, and each variety, remember, has slightly different chemistry. And any time a molecule has different chemistry, it allows it to interact with other molecules slightly differently. Compare that now to the next set of bars off to the right. This is a visual representation of the different types of phospholipid in the plasma membrane of cells that are found in a potato, in a plant, a multicellular plant organism. You can see there are fewer varieties of bars. There are fewer colors of bars here. So there's less variation in the number of types of phospholipid, but we still see the same phospholipids that we saw in the rat liver. We still have the red bar, the phosphatidylethanolamine. We still have the orange bar, the phosphatidylcholine. We still have this blue bar, the phosphatidylinositol. It's just that the bars over here, we're missing some of the variety that we saw in the rat liver. So the membrane that surrounds the cells in a potato are different. The membrane composition is different than the membrane that surrounds the cells in the rat liver. If you go to the next set of bars, this is what you would find in the membrane around an E. coli cell, a bacterial cell of the species E. coli. Over here, this is the variety you would find in a human being. This happens to be a representation of myelin. Over here, this is the variety that you would find in the membrane, the thylakoid membrane of a chloroplast in a spinach cell. And over here, the last grouping, this represents the rat liver again, but this is the membrane that we find inside the mitochondria in rat liver cells. So look, there's even a difference in composition between the plasma membrane of a rat liver cell and the inner mitochondrial membrane of a rat liver cell. From cell to cell to cell, from organism to organism, from domain to domain, membranes differ from each other. They're not all identical. Yes, they are all primarily made of phospholipid and protein, but there are just these slight differences between the phospholipids that give them slight differences in chemistry and slight differences in permeability of those membranes. So one other thing to note about these variations on phospholipids is this naming system. If you haven't picked up on it already, when we name a type of phospholipid, we use this terminology. We call it phosphatidyl X, and the X is whatever small group has been added on to the head of the phospholipid. So if we've added on a choline group, we're going to call it phosphatidylcholine. If we've added on a serine, we're going to call it phosphatidylserine. So some of the most common varieties of phospholipids are listed here: phosphatidylcholine, phosphatidylserine, phosphatidylethanolamine, phosphatidylinositol, phosphatidylglycine. When we modify a phospholipid to have these additional groups attached to the head of the phospholipid, we give that phospholipid slightly different chemistry. And anytime you alter the chemistry of a molecule, you're going to change its function.